All right. Hey, everybody. Today I'm going to be talking about lake trout, Lake Erie, try and focus in on the last 10 years of work. A lot of different projects have come into the last 10 years, and I think now is probably the best time to bring this whole story together. None of this work could have been done without a lot of different people. I'd like to pinpoint these co-authors here for this work, but a lot of other collaborators with this. So just a brief backstory on Lake Erie lake trout. They were extirpated in the 1960s due to overharvesting by commercial fishing. We have habitat degradation, eutrophication, invasive species. Not a good scene, but something not uncommon for the rest of the Great Lakes. Fast forward to the 1970s, we start concerted stocking. Fast forward again, we're at a point now where we have this stocked trophy fishery of lake trout. Both of these are Lake Erie lake trout. I will point out, however, they have adipose fin clips. All of our stocked fish have, like Mark said, a code wire tag inserted at stocking as well as an adipose fin clip. That's going to come back around later, but something I wanted to point out here. So we're talking about the eastern basin of Lake Erie. That's where we have the cold habitat, especially during thermal stratification. It's where we have the deep water. Now this figure here is our cold water gillnet catches over time, and I'll focus first at the far left of this graph. 1985, we had no adult lake trout in our cold water gillnet assessments. The blue line there is age 5 and 6. The gold is age 7 and older. It wasn't until sea lamprey treatment began that we finally started to get fish into those older year classes, or older ages. You fast forward again to the far right side of this figure, and we have a much healthier adult population of lake trout in the lake. Now, another way to look at this is age distribution. You look at 1985, we had no fish getting past age 4. Sea lamprey were having that big of an impact on our stocking. And at 2023, you see we have a really healthy age structure of lake trout in the lake. In the far right corner of the bottom here, the 20 plus year class, we have fish all the way up to age 35. We have a lot of fish that are able to get to older ages, which is great for trying to rehabilitate our lake trout stocks. Another thing that Mark touched on earlier was our lake trout strains. What we also found was that the Lake Champlain strain in gold and the Finger Lake strain in blue are the two strains that have the best survival to adulthood. That's what we've transitioned to stocking now, but making that step was another big process along the way that I'm going to move past fairly quickly. But what I will say, the Lake Champlain and the Finger Lake strain are very similar. The Finger Lake strain are what were originally stocked into Lake Champlain, so they're genetically very close. So let's have a quick review before I dive into the last 10 years. Lake trout extirpated from Lake Erie. It wasn't until sea lamprey control and some concerted stocking that we finally got adult lake trout back into Lake Erie. We had to start stocking the right strains to get a better number of adult lake trout in the population, and we're at a point right now where we have this really healthy age structure in the lake. But during all this time, we still haven't found wild lake trout in Lake Erie. And the first question for us is, we have all these fish, why aren't we seeing any wild reproduction or recruitment? The first thing we point to is habitat, spawning habitat. This is 2 pictures of Brockton Shoal. It's what we thought of as our historical spawning habitat out in Lake Erie. It's out in 50 to 60 feet of water in New York. It's this really large reef complex, beautiful habitat before dry seeded mussels. So the image on the left here is within the 2 years of when sea lamprey treatment began, but it's before we had a buildup of adult lake trout in the system. Once dry seeded mussels showed up on the scene in the late 80s and early 90s, we have a buildup of not only dry seeded mussels, dry seeded mussel hash, we have siltation. It's fully impacted. Today we don't have lake trout congregating on those sites. We have a very healthy age structure of adult fish. They have to be trying to spawn somewhere, but we didn't know where that was. let's see here. This is where acoustic telemetry and the GLaDOS network really came into play. From 2015 to 2019, we tagged 285 adult lake trout. This was all done in the Eastern Basin between Pennsylvania, New York, and Ontario. We also had help from USGS and GLaDOS with the tagging. We tagged these fish at a mixed fishery to try and remove biases of pinpointing where they were going at spawning. So we tagged them during the late spring, early summer, before we had stratification buildup. We tagged these fish in the different jurisdictions, and we let them go and see where they go during the year. These tags were 4 and older tags, so they had a longer lifespan, so we could track these fish over a longer period of time and try and get as much data as we could from these fish. We also set up this near-shore perimeter array around the East Basin, both on the north and south shores, to see where are they going at spawning time. So this video here is going to show movement of those lake trout in November of 2016, or October to December 2016. The black dots are the individual receivers. You see the grid array is what is covering all the center of the lake, and then we have our perimeter array that are the black dots going all around the Eastern Basin. The blue dots are individual fish moving during this time period. So if you could press play, please. So what we see here is most of the fish are in the Eastern Basin. Makes sense, it's where we have the cold habitat. Once we get to this turnover, what you're going to notice is these fish start going every which direction. They're searching for the right habitat to spawn. And once we get over here to the spawning time period in late November, they all start clustering right between the PA and New York border on the southern shore. And when I say most of them, I'm talking the vast majority of these fish all congregate in to this site during that spawning period. So what we did after that point was we set up these tighter fixed arrays during the spawning period in subsequent years, starting in Pennsylvania in 2017, and then Shorehaven Reef was 2019, Van Buren was in 2018. We set up these 4-mile wide arrays, set the receivers close enough together that we could get sub-meter accuracy for these individual fish. So these black circles here are the individual receivers. The red dots are all the individual fish detections. I'm focusing on Shorehaven Reef here. That's where we had the most individual detections. It's where we had the strongest congregations of fish. Right here and here were the 2 strongest congregations from the 3 fixed near-shore arrays that we had set up. So Shorehaven Reef, it's western New York this is 15 to 25 feet of water, roughly, is where this grid was set up. So we're going to look at this another way here with a heat map, with purple being the strongest congregations, and then yellow being, there's still fish present, but it's not where they're spending the majority of their time. And this is overlaying a LiDAR habitat map. So you can see what the habitat looks like, the bathymetry, where those lake trout are congregating. So I'm going to focus in on those 2 congregations of lake trout and take away the heat map. And what you see is those are over habitat breaks. So this is 2019 that we set up this array at Shorehaven Reef. We got the data back in November of 2020, looked and saw those congregations and said, we need to get out there as soon as we can. We went out the next day, and this is what we found. We have this large congregation of lake trout, 7 fish deep. We have fish porpoising all around us during the daytime, other spawning behaviors going on. We have a lake trout with an adipose fin present, which was at least an interesting side note. But most importantly, we have clean, broken cobble. That's something that we didn't think was necessarily possible in Lake Erie at this time. With all the Dreissenid mussel invasion, we thought this would be fully impacted, and we found some clean broken cobble in one section of Shorehaven Reef, and the lake trout found it as well. So this is November of 2020. We have visual observation of these fish clustered together, having some other spawning like activities. So we go back out there in April of 2021, and in short order, we find fry. We send some samples of those off to Chris Wilson up in Ontario, and he comes back and says, yes, those are lake trout. They're Seneca strain lake trout. Okay, this is coming back to the Finger Lakes, Lake Champlain conundrum. We couldn't quite tease out which of those strains they are, that these wild fish are coming from, but it's one of those two, which is a good thing for us to know because we're also stocking those two strains now. I think we have the technology now. We can get at that. We just haven't gotten to that point yet, but we hope we can in the near future. So we went back out again in 2022 and 2023, and we got wild fry again at both of those two congregation points. So this led us, Mark presented this earlier, led us to looking back at our cold water assessment to say, okay, we have some fish that have adipose fins present. We have fish that don't have a coated wire tag. We know the error rates that are involved with that. So we took a harder look, and after accounting for that error rate, the combination of the two, this is what we found, was that there's a very small percentage of wild fish that are being caught in our cold water assessment. Prior to this study and process, we assumed that that was zero. We just weren't seeing it. So after taking that hard look and finding those wild lake trout fry, it helped move the needle that much further along. So this was one of the cleanest parts of work and research that I've been able to be a part of. We were able to get a really nice publication out of this. You can find in the Journal of Great Lakes Research. Every step led to the next logical step, from just taking the assumption away that we knew where we thought they were trying to spawn, let the fish show us where they're going, and being able to follow them and see it came to fruition. Once we got out of the way, the fish showed us where they were trying to go. So we've been able to find wild fry at this reef for 3 years in a row. What are the next steps? Well, we want to continue fry sampling, but we want to go to some of those other smaller congregation sites. There's some in Pennsylvania. We have seen some congregations as well in Ohio. There's fry traps available to go out and try and do some of that sampling to see if we can't find some other spawning habitat and spawning successfully recruited fry in other sections of the lake. We're also working with Dmitry Gorski's lab and Joe Johnson at trying to map and measure some of this habitat in the lake to get a better understanding of what we have to work with out here and what the lake trout have to work with. We're also working with USGS and the University of Buffalo, Dr. Joe Atkinson, to try and get at the hydrodynamics. Why is this particular reef clean when so many other places are filled in with sand and silt and dry seeded mussels? We're also looking at doing some experimental habitat addition and rehabilitation, otherwise cleaning, trying to see if we can clean the reefs that are out there now and how long they might stay clean, or if we put in the rock in the right place, can that rock stay clean for a long enough period of time for it to be viable for lake trout, for a longer period. And this is going on both in New York and Pennsylvania. And finally, we need to continue sea lamprey treatments. Without that, we wouldn't have the adult population of lake trout available to try and get wild lake trout back into Lake Erie. So with that, this was a major collaboration with all of these different organizations. It could not have been done with a lot of different collaborators here. And with that, I'll take any questions.